University of Florida. I am working in Dr. Strekalova's lab in the computational microscopy imaging lab. And today I will talk about the educational implications of the fusion technology. So for a little bit of background, fusion is the functional unit state identification in whole slide images. It's designed to view transcriptomics data in relation to histology through artificial intelligence, or commonly known as AI. Functional tissue units, or FTUs, are segmented through this AI to map the histological features of cells. So the aim of the research at hand is to expand and look into the educational uses of the fusion technology beyond the traditionally considered disciplines. So the research is a little bit more public health focused as in comparison to true wet lab research. So here I have images of the fusion technology. So here we're able to see the actual whole slide images. The yellow structure is one of the whole slide images that we have. And on the right-hand side, we can see the different tools that the fusion technology contains, like the zooming in and out feature, the annotation control feature, and the layers control feature to really be able to toggle whatever a particular person may need from this technology. So the annotations, like I said, are one of the bigger parts of this tool. Right now, the tool is designed around kidney cells, so that's why you'll see the glomeruli, the tubules, the arterioles, and the spots. Hopefully the technology will be expanded past this, but as of now, if you do use the fusion technology, you're able to select what structures you would like to see with the cell. So if you want to just see the tubules, you're able to select the tubule box and look for that, or select everything if you want to see everything. There is also a heat map overlay function where you're able to see immune cells and neural cells and epithelial cells to, again, really tailor it to what a particular person may need. The cell composition and graphics is another large part of the tool. So on the left-hand side, we can see that the fusion technology is able to segment the cell a little bit and be able to pick out the composition of the cell, like the pie chart here. Depicts the different components of the cell. It breaks it down. It can tell you how many tubules and arterioles and everything there are. And on the right-hand side here, like I said, because right now we're really only working with kidney cells, we're able to also input nephron diagrams and what a default cell might look like to be able to compare the structure you're looking for. Of course, a real life nephron on a cell slide looks very different than a lot of the traditionally drawn nephrons that we see in textbooks, but it is included to give individuals, the option to be able to compare what they might see in a textbook to what they might see on a real life histology slide. So for the results, skip the slide. For the methods, right now we are analyzing transcripts from previous interviews with undergraduate biomedical engineering and electrical engineering students. These students were shown a video about the fusion software where they were able to run through it and kind of see everything that I explained on the previous slides. And the students were able to give their inputs in these transcripts based on what they thought the educational implications of the technology might be. So for example, they were directly asked how they believe this software could be used both in context of their classes and out of it, just to kind of pick the brains of the students a little bit and see where they thought it might be applicable. The transcripts were analyzed qualitatively through searching for action verbs. So for example, while reading the transcripts, if we noticed that a student was saying, we can use the fusion technology to understand the cell, we can use the fusion technology to study, we can use the fusion technology to learn in this class, those were picked out, and the overall sentence that the action verb resided within was summarized, and that message was extracted as a code. The Microsoft Word macro was then used to extract all codes to Excel sheets. So as of now, every transcript from these students has a separate Excel sheet with it where the comments are extracted and ordered, and the codes within the Excel sheet are currently being ordered to continue the analysis process and be able to better understand the ideas that the students came to us with. The end goal is to identify these standard codes, which will then be grouped into overarching themes to, again, better understand where the students may see the fusion technology used in the future. So for the results, the project is still in progress and the results are still being generated. Being able to extract the code from the transcripts took a little bit longer than anticipated, and being able to summarize transcripts is also a very long process. But our end goal is that the results will provide a wide range of ideas and educational uses for the fusion technology. As of now, a majority of the students that we interviewed do believe that the fusion technology can be used in undergraduate and graduate contexts, specifically in biology, histology, and pathology courses to gain a better understanding of the material that's being taught in lectures. The interviewees overall believe that adding a fusion component to classes would aid with better understanding the material taught through more traditional means like lectures and textbooks. I know my fellow students on this call can probably attest to going to a lecture and leaving a little bit confused if you're only listening to a professor talk about a certain cell structure and not really being able to visualize how it would look on a real pathology or histology slide. So a lot of students believe that the hands-on approach to learning can help reinforce the information being taught in these more traditional settings and really help foster a better learning environment in general in different courses. So in conclusion, the results of the project will guide the future development and implementation of the fusion technology. The results will be used for the development of an online Canvas course to educate new users on how to use the tool. This can range from undergraduate students to graduate students and beyond. Of course, a technology like this has many applications. So the goal is to be able to develop different Canvas courses to be able to teach people depending on what they may need this technology for. The applications of technology and AI in healthcare and biomedical research are growing exponentially, as we may know, especially in today's day and age. So research like this is really important, especially considering the intersection between public health, education, and technology, which continues to be a vital area for future research. And I believe that the project at hand will continue to contribute to this important field. But at this time, I'm more than happy to take any questions or open the floor for discussion.